My name is Niji. I'm a graduate student working in Professor Alexander van Oudenaarden's lab in the biology department. My research is trying to study cell signaling pathways, and specifically the Wnt signaling pathway. Wnt signaling plays a very major role in mediating coordinated behavior between cells, and perturbation of Wnt signaling has been shown to play to be a key contributor to many diseases such as cancer, neurodegenerative diseases. So one fundamental problem is just to understand how Wnt signaling specifies different biological outcomes in a pool of cells that initially start with similar cell fate. So you can imagine, for example, in stem cell, you actually start with cells that are very homogeneous and they all have similar cell fate and cellular environment, but somehow through this signaling activity between the cells, eventually the cells take on different cell fates. So we use this specific event that happens in the worm to try to figure out how two cells that initially are very similar can utilize Wnt signaling to determine two different cellular behavior. So in this image, we're only showing one out of the two cells that were interesting. Actually, on the other side of the worm, there's almost a mirror symmetry of this side of the worm. There's another cell that's born at exactly the same place, but does the opposite behavior of this cell. While this cell is trying to migrate towards the tail, the other cell migrates towards the head. So we wanted to figure out what is different between these two cells, and what mechanism is Wnt signaling using to produce this two opposite behavior in the two cells that presumably have very similar environment. So first, I thought I really appreciated the beauty of the C. elegans as a model organism. It's a very simple organism that consists of less than 1,000 cells in total. I also like the image because of the three basic color that's used in this image: red, green, and blue. Aesthetically, it's a beautiful combination. I also love the shape of this worm. So C. elegans is actually a soil worm that goes in sinusoidal shape. So it basically curls around and moves in this sine wave. So it's actually hard to capture it when it's curled up, kind of in a resting state. So this newborn worm actually happens to be in a nice circular state, and it's kind of inactive. Presents a very nice symmetry.